Myself, Dr. Savita Ekka, Department of Plant Pathology from Birsa Agriculture University. Today, I am going to deliver lecture on the topic History of Plant Pathology with special reference to Indian work. In ancient period, Greek philosopher Theophrastus was the first person who studied and write about the diseases of tree, cereals and legumes in his book Inquiry into Plant. In India, the information of plant diseases is available in ancient literature such as Rig Veda, Atharveda, Vishnu Puran, Agni Puran, etc. Vriksha Ayurveda by Surpala in ancient India is the first book in which a lots of information on plant diseases is available. During 17th century, the invention of microscope gave vision to unknown world of microscope, and a Dutch worker Leeuwenhoek who construct first compound microscope in the year 1617 and discovered bacteria in this microscope. During pre-modern period, a scientist Pier Antonio Micheli, he was an Italian botanist and founder and father of mycology. He was a person who observed fungal spore for the first time and conducted many spore germination studies by growing fungal organism on freshly cut pieces of melon and pear. He was the person who observed cystidia on laminar edge or hymenonium layer of auricles. During the year 1729, he published a book Nova Plantarum Genera, in which he gave description about 1900 species in Latin, out of which 900 were fungi. The important genera in his book are Aspergillus niger, Botrytis species, Polyporus species, etc. Next to Micheli, Matthew Tillet, a French botanist, published a paper on burnt or stinking smut of wheat in the year 1755. By well-planned experiment, he proved that wheat seed that contained black powder on their surface produced more damage than clean seed. He emphasized that burnt was an infectious disease and caused by some toxin present in the black powder. He reported that the chemical treatment of wheat seed with common salt and lime inhibit the contagious activity of the pathogen. Next is Cornelius Linnaeus. Cornelius Linnaeus is a versatile taxonomist and considered as father of taxonomy. He introduced binomial nomenclature. Binomial nomenclature is scientific method of naming plant, animal, microorganism composed of two names consisting of genus and species. He wrote a book Species Plantarum in the year 1753, is considered for nomenclature of all fungi except fossils. He guided two students, Christian Hendrik Persson and Elias Magnus Fries. They applied binomial system of nomenclature to fungi and gave firm footing to the taxonomy of fungi. Magnus Fries, regarded as Linnaeus of mycology, he wrote a book Systema Mycologicum, is the chief starting point for nomenclature of fungi. Hendrik Persson wrote a book Synopsis Methodica Fungorum, is the chief starting point for nomenclature of rust fungus come to the order Uridinales and Astilaginales, that is smut fungus and Gastromycetes. In modern era, Isaac Benedict Prevost, a French botanist, first experimentally proved that burnt of wheat was caused by a microorganism. That microorganism is Tilletia caries. Tilletia is the causal organism of burnt of wheat. He also discovered the life cycle of fungus and showed that the solution containing copper sulfate prevented the germination of the burnt spore and it is used for control of one disease in this time. He also mentioned the fungicidal and fungistatic properties of chemicals. Now, the history begins with potato famines. Potato famine, that is Irish famine, which was a tragic event in human history. This disease caused great Irish famine during the year 1845 and 46, and due to these famine, over 1 million of people died and 1.5 million people were migrated. In the year 1840s, Tulasne brothers, these Tulasne brothers are Louis René Tulasne and Charles Tulasne, at Museum of Natural History in Paris begins extensive morphological studies of fungi. They made illustrated drawing of rust, smut and ascomycetes. Louis René Tulasne is known as reconstructor of mycology. Next one is Henrik Anton Debery. Anton Debery is considered as father of modern mycology and plant pathology. He was the scientist who indicated the nature of obligate and facultative parasite. In the year 1861, he finally proved that Irish famine caused by late blight of potato is caused by Phytophthora infestans. That is, Phytophthora infestans is the causal agent of late blight of potato. He also discovered heterogeneous nature of rust and life cycle of many downy mildew fungi. Working with Sclerotinia sclerotiarum, this pathogen is coming from white rot and wilt of vegetable, opened entirely new field of investigation, that is physiology of parasitism. The another contribution of Debery was the training he imparting to the students. Famous among them are Voronin of Russia, Brefield of Germany, Millardet of France, Ward of England and Fisher of Switzerland. Julius Kuhn was contemporary of Debery, published first textbook for farmers in the year 1858. The title of textbook is The Diseases of Cultivated Crops, Their Causes and Their Control. Next, from the Kuhn, Brefield. Oscar Brefield of Germany in the year 1875 developed many modern techniques of inoculation and growing microorganisms in pure culture. Fungicidal era of plant pathology started with P.A. Millardet in the year 1878 with the discovery of Bordeaux mixture. Bordeaux mixture is the combination of copper sulfate and lime, and this mixture is used for the control of downy mildew of grapevine. Orton and Biffen mentioned a pioneer in the field of resistant breeding. In the year 1905, Biffen described inheritance of resistance to yellow rust, and in the year 1909, Orton distinguished disease resistant from disease escape and disease tolerance. Erickson in the year 1894 discovered phenomenon of variability among the fungi. Flor explained host-parasite interaction in flux rust by gene for gene hypothesis. According to the hypothesis, susceptibility of disease depends upon the compatibility of gene in host and the pathogen. Next to the Flor, Vanderplanck suggested two kinds of resistant. One resistant is controlled by few major genes, but it is strong and race specific and termed as vertical resistance, and another resistance is determined by many minor genes which is weaker but effective against all races of pathogen is called horizontal resistance. The bacteria can act as a specific infectious agent of disease was first made in the animal through the study of anthrax. And Louis Pasteur and Robert Koch in the year 1876 proved that anthrax disease of cattle is caused by specific bacterium. Robert Koch, considered as father of bacteriology, he introduced staining technique and obtained bacteria in pure culture. He proposed famous Koch postulates which is used in pathogenicity test, and the criteria of pathogenicity test is, first criteria is constant association of pathogen, second is isolation of pathogen in pure culture. Isolation means separation of pathogen from the host. Third criteria of Koch postulates is identification of isolated pathogen. Fourth criteria is inoculation. Inoculation means transfer of pathogen into host of isolated pathogen, and last criteria is re-isolation of inoculated pathogens. E.F. Smith reported series of paper on cucurbit wilt, brown rot of solanaceous vegetable, angular leaf spot of cotton and blight of bean, and considered as father of bacteriology. The role of nematode was first reported by Needham in the year 1743. He discovered first phytonematode, that is wheat gall nematode. Its scientific name is Anguina tritici. C. Devine discovered life cycle of this wheat gall nematode in the year 1857. Berkeley is the scientist who discovered second phytonematode, that is root knot nematode, and the scientific name of root knot nematode is Mileidogyne incognita. Bob in the year 1931 studied structure of many nematode and classified them. He developed many techniques for study of nematode and coined the term nematology. Next, role of viruses in plant pathology. Earliest reliable record of viral diseases are flower color breaking of tulip, also known as tulip mania, and degeneration disease of potato. Adolf Edward Meyer in the year 1882 first transmitted tobacco mosaic disease and reported that the tobacco mosaic disease was neither due to microorganism nor due to nutritional imbalance. He demonstrated contagious nature of causal organism by artificial inoculum and proposed the term mosaic. Mosaic means pattern of light and dark green areas. Next is Ivanovsky, Ivanovsky repeated the work of Meyer by passing the sap of infected tobacco leaves through Chamberlain bacteria-proof filter and concluded that tobacco mosaic disease was caused by toxin secreted by bacteria or smaller bacteria. Martinus William Byring, father of virologists, repeated the work of Meyer and Ivanovsky and stated that tobacco mosaic disease was neither caused by bacteria nor by toxins secreted by bacteria but caused by a contagious poisonous fluid and proposed the term contagium vivum fluidum hypothesis. He used the term virus, which is derived from Latin word virus means poisonous. Holm listed 129 plant viruses and developed local lesion assay for quantification of viruses. Chemical era of virology started with Stanley. He isolated tobacco mosaic virus in paracrystalline form and concluded that tobacco mosaic virus is a autocatalytic protein which multiply only in the presence of living host. By his work, he was awarded with Nobel Prize in the year 1935. Borden and Peary in the year 1936 proposed that tobacco mosaic virus is nucleoprotein in nature, which means it consists of nucleic acid and protein. Koschei, Puffinkuch and Ruska in the year 1939 obtained first electron microbe of tobacco mosaic virus. William and Wyckoff in the year 1944 subjected shadowing with heavy metal in order to improve the contrast, and it is known as shadow casting. Braque in the year 1951 reported density gradient centrifugation for purification of plant virus. Girer and Schramm in the year 1956 conducted experiment on reconstruction and fractionation of tobacco mosaic virus and reported that infectivity is only the property of nucleic acid and core protein play only a protective role. Casanis in the year 1962 gave the theory of satellitism. Satellite viruses are the smallest viruses, spherical in shape, 17 nanometer in diameter and occur only in the presence of tobacco necrotic virus. Miura, Kimura and Suzuki in the year 1966 reported that double standard virus containing double standard RNA as a genetic material, and two important viruses which contain double standard RNA are rice dwarf virus, wound tumor virus. Shepherd et al. In the year 1968 reported that virus containing double standard DNA as a genetic material, and the example of double standard DNA viruses are cauliflower mosaic virus, Dahlia mosaic virus. Wöhler et al. In the year 1976, Clark and Adam in 1977 gave ELISA. ELISA is the enzyme-linked immunosorbent assay technique for detection and identification of mycoplasma, bacteria and some fungi. Denner discovered new kinds of pathogen, that is viroid, in plants, potato spindle diseases. Viroid is a small, naked, single standard circular molecules of infectious RNA having low molecular weight. Doyo et al. In the year 1967 observed mycoplasma-like bodies present in the phloem of plant suffering from yellow diseases. Some another mycoplasmal diseases are potato phyllodes, marginal inflorescence, this is also disease of potato, little leaf of brinjal, grassy shoot of sugarcane. Ishii et al. In the year 1967 reported that mycoplasma-like bodies are sensitive with tetracycline. Davis et al. In the year 1972 observed a motile helical wall-less organism associated with corn stunt disease, which he called spiroplasma. Some spiroplasmal diseases are citrus stub-borne, aster yellows, rice yellow dwarf. Then plant pathology in India. D.D. Cummingham and A. Brackley started the identification of fungi in India, and the plant pathology started as a university subject in the year 1930 at Chennai, Allahabad and Lucknow. K.R. Kirtikar was the Indian scientist who collect and identified fungi in the year 1967. Imperial Agriculture Research Institute was established at Pusa, Samastipur, Bihar, but after earthquake in the year 1934, this institute was shifted to Delhi and recognized as Indian Agriculture Research Institute, that is IARI. Edwin John Butler is considered as father of Indian plant pathology. He stayed about 20 years and trained several Indian scientists, working with Pythium and Phytophthora, and wrote a monograph on Pithecious and allied fungi. He wrote a classics, Fungi and Diseases in Plant. This was published from Kolkata in the year 1918. J.F. Dastoor is the colleague of Edwin John Butler. He was the first scientist who was credited for the study of fungi and plant diseases with special reference of Phytophthora causing diseases in castor and potato. Dastoor was internationally known for the establishment of species, that is Phytophthora parasitica. Phytophthora parasitica is the causal organism of blight of castor. Next is K.C. Mehta. K.C. Mehta discovered the disease cycle of cereal rust in India. B.B. Mundkar started the work of cotton wilt through vertical resistance. He identified and classified many Indian smut fungi. He established Indian Phytopathological Society in the year 1948 with its journal Indian Phytopathology. He wrote a textbook, Fungi and Plant Diseases. J.C. Luthra developed solar heat treatment on wheat seed for control of loose smut of wheat. R. Prasad worked on rust and identified their physiological races. He discovered the life cycle of linseed rust. S.N. Das Gupta and P.S. Sadasivan established biochemistry of host-parasite relationship and gave concept of vivotoxin. M.G. Thirumalachar introduced antibiotic in plant disease control. R.S. Singh, a professor and head of plant pathology in Pantnagar, working on biological control and ecology of soil-borne diseases. He wrote a textbook, Plant Diseases. Y.L. Lane investigate cause and cure of Khaira disease of rice. This is all about the history of plant pathology in your course. Thank you.